Hello, everyone. Thank you for joining us for a live look at the restoration of mesophotic and deep benthic communities in the Gulf of Mexico. My name is Sasha Francis, and I'm the Gulf of Mexico Restoration Education and Outreach Manager for the National Marine Sanctuary Foundation, one of the many partners helping to make this restoration work possible. Last year, we tested an awesome setup on board our ships that allow us to live stream from the NOAA ship Nancy Foster. And now we're back on the Nancy Foster again this season with more opportunities to connect with folks at sea. We're really excited to have you join us to get a sneak peek of science at sea and see what it's like to live on a ship for a few weeks at a time. So as we go, please share any questions or comments that come to mind in the chat, and we'll pause for questions throughout as we meet the folks on board. We've worked with partners at the University of Rhode Island's Inner Space Center to install Starlink systems on the ship that allow the folks on board to have a strong enough internet connection to stream data, audio, and video to us on shore. This allows the science team on board to collaborate with team members on shore in real time, opening up opportunities for scientific collaboration as well. All right. In a few minutes, we're going to connect with our team on board. But first, I'm going to give you a quick little overview along with some cool video clips to talk about why they're out there in the first place in the Gulf of Mexico and what it is they are seeing. So by now, you've probably heard the phrase mesophotic and deep benthic communities. It is a mouthful, but it's the scientific way of referring to the ecosystems, the habitats that you can find on the seafloor at depths below 50 meters from the surface, or about 60 feet and beyond. We're even working at depths past a mile below the surface. So let's dive a little deeper into what these communities are, why they're so important, and why our scientists are currently on a ship in the Gulf of Mexico working to learn more about them and restore them. So as you go deeper into the ocean, like the underwater robot you here see, a remotely operated vehicle, or ROV, less and less sunlight is able to penetrate the water. Eventually, you reach the mesophotic zone, which literally means middle light. It's sometimes referred to as the twilight zone. As you travel even deeper, you'll eventually reach a point where there's no sunlight at all. In addition to this change in light, the water also gets colder and there's a higher pressure. Despite the low light, cold temperatures, and high pressure environment, there are incredible ecosystems that call the seafloor at these depths home. Colorful corals and sponges are the foundation of these ecosystems, providing habitat for the many fish and invertebrates you see here. It's much more colorful than you think, I bet. These deep sea ecosystems are found all over the world, including in the Gulf of Mexico, which is where all this beautiful video footage is from. But there's still so much we have to learn about the deep sea. Exploring these depths can be difficult and requires help from expensive, highly technical equipment, some of which we'll learn more about from the science team today. However, we do know that the deep sea is the largest habitat on Earth. The number of species and abundance of life that depends on these deep sea habitats rivals the colorful coral reefs found in shallow coastal waters or tropical rainforests on land. Unfortunately, the Deepwater Horizon oil spill in 2010 impacted these important deep sea ecosystems in the Gulf of Mexico. And restoration is challenging because of how far from shore and how deep they are, because of all the things we still have to learn about them. As a result, 4 restoration projects are currently underway to help us better understand and restore these ecosystems. So we have 4 different projects that are working together, like I mentioned. And one of them is developing better maps of the seafloor in the Gulf of Mexico, so we know where these coral habitats are found and where to go to look for them. Some other projects we have here, the Habitat Assessment and Evaluation Project, learning more about the species that live in these habitats and how they may be connected across the Gulf of Mexico. Testing different methods to help corals grow and reproduce in the wild and in labs in order to help replace the corals that were injured by the oil spill. And working to reduce the threats that these ecosystems face and connect this restoration work to the public and much more, which is my role on the Active Management and Protection team. This work is made possible by a number of different partners, including a large number of academic institutions like universities and non-governmental organizations like the National Marine Sanctuary Foundation, and 3 federal partners, the National Oceanic and Atmospheric Administration, or NOAA, the U.S. Geological Survey, or USGS, and the Bureau of Ocean Energy Management, or BOEM. So without further ado, let's connect to some folks on the NOAA ship Nancy Foster, the really exciting part. So just a note, you might see pauses in the video at times. That's okay. It's just how things go when you're connecting with folks on a ship in the ocean. So let's see who we have here. Looks like we have a view of the deck. Oh, hello. We have multiple friends. Hi. Hi, Alexis. You want to introduce yourself and tell everybody a little bit about what you do on board?
Sure. Hi, I am Alexis Weinig. I work with USGS at the Eastern Ecological Science Center in Carneysville, West Virginia. I think we have Miss Rain's class joining us today from West Virginia also, which is awesome. So I am a biologist with USGS, and our lab focuses mostly on coral genetics and then also environmental DNA or eDNA. So we are collecting seawater when we're out here and filtering that seawater to get little tiny particles of DNA so that we can have a better understanding of what animals might be in the area of that water. I'm also acting as the lead scientist on this expedition, so that entails organizing all of the science that's going on board and making sure that we're working well with our NOAA Corps officers who are driving the ship, as well as our deck team and other ship staff, to make sure that we're performing all of our operations safely and in the time that we have to do them. So that's my main role.
Awesome. We'll pass it out to the folks on deck.
Hi, I'm Jenny McLean. I am also a biologist with the U.S. Geological Survey, and with me is my lab mate.
Hi, I'm Maria Gorgas, also a biologist with the USGS.
And Maria and I are leading up the sediment collection activities. So we're taking push core samples, and a suite of different analyses are being performed on those sediment cores. We look at the geochemical properties. We look at the sediment types, whether it's clay, sand. We look at the animals present within those, looking at diversity and abundance. In addition to that, our lab also will take isotope samples. So we take tissue samples and we'll look at the carbon and nitrogen ratios to look at food web structure in these deep sea habitats. And then we also filter water to get particulate organic matter, or POM, which is often the baseline for sources of food for the food web.
Awesome. So when you're taking sediment samples, how do you get those if it's like a mile underwater?
So as you saw earlier, the ROV, or the remotely operated vehicle, will go down into the water, and it's tethered to the ship, so it kind of stays with us, but it's controlled by an ROV team. And once it gets down to the bottom, we have these plastic tubes. They're called push cores, and they are essentially pushed down into the sand, and they come back up and close and they're filled with sand after that. And then they get put back into a little basket on the ROV, and once the dive is over, the ROV brings them back up to us, and then it's our turn to take care of them and go through all of the different cores that we have for that dive.
Oh, so you cut little slices of the mud basically and look for different things inside. Awesome. What are some of the little creatures you might find in a mud sample? Oh, I think you might be muted with this video playing. Hang on a second. We see some footage of the mud being processed here. I think we might be able to hear you now. Oh, this is a great video, watching those little slices that I just mentioned being taken from those cores and processed right in the lab, right on board. Awesome. So they take smaller samples in tubes of those larger cores that you saw taken into little slices so they can analyze different things. Okay, I think we can hear you now.
Okay, great. So we have a lot of different animals that pop up in our samples. When we're in the lab, I'm actually sorting through them. So we dye the samples with something called rose bengal, and it makes it so that the organisms that were once living will show up very bright pink, so they're easy to find. And we've had a lot of different things. There's all different types of crustaceans. We have a lot of annelids. So those are worms. They have cheti, which are like almost like spines that come out of them, but they're very fun to look at, and there's so many different types of them. My personal favorite are the brittle stars. So they're very similar to sea stars, but they kind of have like a central disc and then very skinny arms most of the time that come out. And they're just so beautiful. I have such a good time when I'm in the lab taking photos of everything through the microscope. So I have a large stock of all of our invertebrates if you guys would like to see those at some point. So
I love that. I didn't know you would find brittle stars in the mud. We see them often on the corals we see on the ROV footage. So that's very cool. Okay, question for, I know this came up as I was talking to people about these projects. Comparing, mud samples that you find closer to the wellhead or the impact zone, comparing those to, healthy sites that weren't injured by the oil spill. Have you seen any differences yet, or is that what we're working on finding out now?
So it's definitely an ongoing process. We're very fortunate that in some of these areas we had sampled pre-oil spill, so we have some data before that situation had occurred. But we do notice a difference. So early on in the restoration process, looking at them, we would actually see globs of oil in the sediment. We see that a little less as that oil has been broken down over time. But also with that, the sediment cores were a lot more anoxic with little oxygen. And so we'd see a presence of less animals. So the diversity and abundance was lower than what we're seeing at some of the reference sites. But again, this is an ongoing process, and we're trying to understand how the communities change with these types of impacts.
Awesome. Thank you. And I know that's really a big, important part of this work and these restoration projects is what's going on beneath the corals that we're seeing. And it's really important to the whole ecosystem as a whole. Awesome. And Alexis, you're in the lab. Is that where you are right now?
Yeah, so I'm sitting in the wet lab, which is where some of those photos and video of Jenny and Maria processing, that's where all of that would happen, would be right in here. So this is a space where all of our scientists can have, certain stations set up or equipment that they might need for once we're back on deck with the ROV or with the CTD, which is another piece of equipment that we use. So that's the wet lab.
Awesome. What does the CTD do? I don't think we talked about that yet.
Yeah, so a CTD stands for connectivity, temperature, and depth. So it's actually a sensor that can measure those parameters in seawater. And so it's attached to what we call a rosette. So that's like a large carousel that has, there it is on the screen, that has water samplers attached to it. So we can take 12 distinct water samples all the way down from the seafloor up to the surface. And we do that before we do the ROV dives so that we can have an idea of what the water column parameters look like, and then also so that we can collect water samples for eDNA, for nutrients, so what type of nutrients are in the water, and then like Jenny said, for POM, so particulate organic matter, which allows us to better understand the food web. And our lab at USGS has been a part of a collaborative team to develop this filtration process where we can bring our filters and our peristaltic pumps out to the CTD and do this on deck, which allows us to process our samples much more quickly than if we were to bring them into the lab, which can be really important for the rate of DNA degradation that can happen between when you collect the sample and when you're actually able to process it and freeze it. And so you see here in this video, we have myself and Nina Ramos, who's from the Smithsonian National Museum of Natural History, processing the water on deck and collecting those samples. And it's a really useful, CTDs are super useful across the field of oceanography. So we're using them for this particular use, but they have they're very important pieces of equipment for all types of oceanographic work.
Now I noticed, and we have some students on the line here, I'm sure. I noticed you had hair clips
Yeah.
holding. And I want to just note here that all the scientists we're talking to are women. We have a lot of women on board, and I'm like, is that something y'all figured out? Was the hair clips were awesome for holding the water lines?
Yeah, we needed something that could hold those many different, you could see there's a lot of tubing, and so we needed something that could hold things in place, and it turned out that on one of our earlier expeditions, we had some hair clips, and so we started using those, and then we just started buying them in bulk to send them on all of our trips, because, you know, you gotta make do with what you have.
That's genius. Sometimes the best, tool for the job is something you wouldn't expect, and I love that
Yeah,
you also have spare hair clips, so when you're doing all the science, you can pull your hair back. Oh, it looks like
yeah,
it looks like we have a little tour of the ship going through, so we'll take a moment, and we'll come right back to y'all with some questions. Let's see what's going on here. We might actually see
okay.
Alexis on camera from this angle. Oh, wow. Hey, there she is.
Yeah,
So,
so if you
go
want, I
ahead.
can talk a little bit while
Yeah.
Jess walks around and gives us a view of what we're seeing. So she's currently in the wet lab where I am, but she's showing just this area where we have our one of our filtration setups and then the sinks. She's coming around. We have these benches where Jenny and Maria process the sediment, all set up for our next operation. So one of the things you may have noticed from Jenny and Maria being on the back deck is that they are not moving very much and that there's land behind them. And that's because we have been dealing with some really rough weather here in the Gulf of Mexico. So we're currently in port in Pascagoula, Mississippi, to so that we aren't in a dangerous situation. And so we were avoiding a really bad weather storm, and we will be headed back out to work this evening. And so she's in the dry lab now, which is a space that's mostly computers and really all the technology that is behind what is happening on ship. And so there's a system for mapping the seafloor that is in that room. There is the surveys tech, which is the people that run the CTD and make sure all of that is operating and all of those sensors are working. And then you saw a couple of our other scientists in there that are just working while we're here in port. And then I
So
think, go ahead.
I was just gonna say, oh, this is my favorite part is opening this giant door.
So
So
all
she's
the
gonna
doors are watertight. Just to
yes.
quickly, like, we have you have to open and close them so that it's if you were in a situation where you needed to have no water into the center of the ship. Those doors are for that purpose, and it's very important when you're moving through the ship that you continue to open and close those correctly and safely because they're super heavy.
Yeah, that's just why it's so impressive when they open them with one hand. I'm like, "Yes, you must be so strong." All right, it looks like we have a couple of questions coming in. Oh, here's the, sediment cores
Yeah,
giving
so
their
those are
amazing
some
advice.
cleaned sediment cores that are ready and waiting for their adventure down to the seafloor,
They're
hopefully
like,
tomorrow.
can we go back now?" Oh, and then
Yeah,
we have our, control van, which is where the ROV pilots will operate with a bunch of screens to see what's going on, from Pelagic. We have their ROV
yeah,
Odysseus on board, which is what you see behind, Jenny there.
yeah.
Awesome.
So this is a room where when the ROV is in the water, we're able to work with the ROV pilots who would sit in that front row, and they're able to control and drive, use the mechanical arms that are on the ROV, and this gives them many perspectives. So there's a lot of screens so that you can see the front, what the ROV is seeing in its front, what the ROV is seeing in its back. You could see it from the ship's perspective. So on a sonar, where is it in relation to the ship at the surface? Because all of that is important for safely operating the vehicle.
And it's kind of driven like a video game controller, right?
Yeah,
Has
it's very
joysticks,
similar to
buttons. Yeah.
yeah, it's very similar to something you might play if you ever play an underwater game where you're kind of driving around. It looks similar to that. It's just real life.
All right, we got some questions coming in. They said there's a question about eDNA. Can we see that question? All right. Could Alexis explain what eDNA is and what they use it for? And that looks
Sure.
like our our USGS partner school is asking.
Yeah, hi Brenna and hi Mrs. Rain's class. So we eDNA stands for the term environmental DNA. So all of every organism on Earth has particles of DNA that make up its whole body, right? And so what happens is every organism is constantly shedding small particles of DNA. We are when you shed skin cells or hair cells, as well as all the marine animals that we're interested in studying. And so when the marine animals shed that DNA, it goes into the water and it floats around for a while before it would degrade and no longer be recognizable. And so what we're doing with eDNA is we're trying to collect that DNA that's floating around in the water, and we are then able to extract the DNA. So take that and turn it into a computational sequence. So a sequence of letters that tell us specifically what type of organism that is. And so then after we have that DNA back in the lab, we use a computer to process what animals might be in certain areas, and we can compare the different sites and the different depths that we're sampling to understand how those ecosystems might be changing, or what animals might be there constantly, to where if we had another impact like Deepwater Horizon, we could have an idea of if those animals are still there or if that community is changing with that impact.
Awesome. Thank you for explaining that. That was helpful for me to learn, actually, too. All right. We'll see if we have any other questions popping up on the screen in a moment. Okay. We have one from Rebecca Thompson. Do you ever take samples of vertebrates in the water column? Does the ROV have that capability?
Yeah, that's a great question. The ROV that we're working with does have that capability. So they have what we call a suction sampler. So it's essentially like a vacuum tube that they can control with the manipulator arm. Jess is showing that tube right now. And it goes to a chamber. There's 5 chambers within the ROV's body. So we could collect animals that are either suspended in the water column or more difficult to collect with just the arm, like squat lobsters or crabs can be really tricky. So we can use that suction to try and collect those animals. We don't normally collect vertebrates or fish like that because they're too fast for us. But sometimes we can get invertebrates like jellyfish or siphonophores that are in the water column and bring those up to the surface.
Awesome. And I heard you guys saw something cool on the ROV screen the other day. And I think that there's a video clip of everybody watching that happen. I don't know if we can pull that up, but I heard you saw an octopus and that it was pretty exciting because it had been a rough time so far with the weather. But what is your favorite creature you've seen so far?
Yeah, so here's our video of our first octopus encounter on this expedition. So we had a dive where we weren't seeing a lot of biology. So there's often a lot of space where the biology exists in the sediment, like Jenny and Maria were talking about. Oh, there's all of us. I think this is when we first see it. But what this octopus was just a fun sighting for us because we hadn't really been seeing a whole lot that was what we call megafauna, or exciting bigger biology, but we saw this little guy hanging out on the seafloor and visit him for a little while with our camera. We don't collect them. They're too big and they're not on the collection list, but it's always fun to see them on the seafloor.
Yeah, I have seen a Dumbo octopus on the screen once, and I was like, oh my God, that's incredible.
Yeah.
Awesome. What do y'all like to get to eat when you're on board? I know this is a question we get often. So how on this particular mission, what have you eaten that's been your favorite thing? I know life at sea can be a little bit rough, but I want to highlight the good parts of it, aside from the cool octopuses and animals.
Yeah, I'll quickly just mention, I'll start and then I'll pass to Jenny and Maria. They can give their opinions. We have an amazing chef and steward on board. So when we're on board, there's a staff to create 3 meals a day, and they feed everybody in the ship, which is over like 40 people. So it's a really big effort that they're doing every day, but the food is amazing. I always get disappointed when I go home after this ship because I never cook as well as they do. So that's my general opinion, and we're super fortunate to have them with us. But maybe Jenny and Maria have their favorite foods we've had so far.
Yeah, so everything has been fantastic. I struggled to eat a little bit in the beginning with some seasickness, but once I got past that, I was loving it. I think my favorite things so far have been the cabbage rolls because I can't really get that very often at home. And they also made lamb chops one night, which was incredible. Absolutely amazing. Like fine dining. It's so nice.
Yeah, I was a big fan of the chicken coconut curry that we had one night. And Bob had come up with a creation called hamburger soup. And I wasn't sure about it, but you had to sample it and it was phenomenal. And I told him I'm going to need that recipe so I can make it at home.
I would also be skeptical of something called hamburger soup. I know we just got a sneak peek of the ice cream cooler, which is my favorite thing to see on board. It's because I know there's even desserts that you have to choose from, which is awesome. Great. So do we have any other questions popping up in the chat? We'll check that out. Oh, and we're back on our ship tour. And oh, Fania's in there. And I know Fania's been working on some coral imaging. So let's pause for a moment while we try to find some questions and say hi. How you doing? What's going on in here?
Hi everyone, it's going really well. And yeah, I have some photos if you wanted to look at. So basically what we've been doing here in the Gulf of Mexico is going back to these monitoring sites every year since the Deepwater Horizon oil spill, like changes in coral health and growth, especially at sites that had been impacted by the spill. And to do that, we use remotely operated submersibles and a digital still camera handheld by the arm of the ROV to take these photos. And these are a few examples of the photos that we took in the Gulf of Mexico. So that's a species of coral that we call the bubblegum coral because of its color. And it looks a little bit like chewing gum. So that's pretty cool coral. And these are some of the corals that have been impacted by the spill. And just to show you an example of time series, so you have here a photo from 2017. That's a photo from 2022. And that's the photo of the same coral that we took last week, previously in the Gulf of Mexico at one of the impacted sites. And that's again another example of impacted coral, 2017, 2022, and a photo that we took during this expedition. And that's
So
just
it
another
looks
example of these really
amazing.
high resolution photos that we take.
It looks like it's got more color back. Is that correct? Since 2017? Or is that just my trick of the light on the screen?
No, it does. So if you look at this coral, for instance, that one is very impacted. So the yellow parts are basically the healthy parts, and then the brownish grayish color is the damaged. So that's basically the skeleton of the coral. It doesn't have tissue anymore. And so these parts that were really damaged just fell off. And so it does look like that some of the branches actually look healthier, but that coral is also getting smaller because the damaged parts are just breaking off.
Oh, okay. So I know I got a question from a colleague. Their Mr. Salinger's fourth period class, aquatic science class, they want to know what happens when coral gets physically damaged or broken. So when you're looking at those images, what is it, when you're seeing the injury, what has actually happened to the coral?
So it's really hard to tell how the oil affected the health of these corals, but what we see is that some branches, for instance, are losing tissue. So we have the skeleton of the coral that is exposed and tends to be overgrown by other organisms. We have an example here where you have this purple organism. So that's actually a different type of coral, which is a parasite overgrowing the damaged branches. And the other thing that happens is that sometimes the branches are just going to fall off when they're very impacted. And that's an issue because these corals grow very slowly. We're talking about sometimes less than a centimeter growth per year. So if an entire branch falls off, then it's going to take a very long time for that branch to grow back to its original size. And so that's why we've been observing, and that's still ongoing right now, although the damage happened over 10 years ago now.
Yeah, and when it actually happened and the oil was covering the polyps too, that would prevent the coral from eating. Since the corals, especially the deeper ones, don't have the zooxanthellae, they can't create their own food. They have to catch their food. So they wouldn't actually physically be able to do that, I assume, when the oil coverage is on top of the polyps.
That's right.
Well, thank you for sharing that. Those images are amazing. And that's some of the great imagery we can get with those underwater robots, the ROVs, being down at those depths, which is much deeper than recreational divers can go. So it's really important that we have this equipment. All right. Do we have any other questions here from the chat. Oh, there's my favorite dog in the window. All right, we have a question from Rebecca Thompson. One of my students wants to know if you guys get seasick or are marine scientists immune to that? Good question. I'll take that over to Alexis. Do you want to answer that first?
Yeah, I'll start out. So some people do get seasick. We are definitely not immune to seasickness. So normally, if you are prone to seasickness or it's your first time sailing on a ship, we recommend that you take a seasickness or a motion sickness medication for the first couple days at least, and then normally your body will adjust to the movements of the ship. But if we get really rough seas, kind of like what we're hiding from today, that normally can take most people down, where you want to either be outside or in your bed riding it out. But I think that Jenny and Maria might have their own experiences to share also.
So I'm actually a very fortunate person that I guess I got the good genes, and I have yet to get seasick. So I fortunately I don't have to take any medication, and I handle the seas very well. It doesn't mean that different conditions will happen, and I will. So I always bring medication as a preventative just in case. But I fortunately have not had to deal with that.
Unfortunately, I have. This was my first at-sea expedition, so I've been kind of experimenting, trying to figure out which medication works better for me, what things I can do to kind of mitigate the effects or limit it. So I found that going down into the lounge, basically like the basement of the ship, has been pretty effective for me. Laying down is good because I feel like I don't have to throw up, but I can't really do a whole lot, so it's not the best solution. But you kind of just have to work with it and figure out what you can do and maybe just kind of hope for it to pass. And it's not a whole lot of options, but you do what you can.
Do your best. Yeah, do your best. All right, let's take a peek here at where Jess is with the mobile cam for a moment. Looks like she's out by a poolside. What's going on out here?
She is on what we call the steel beach. So this is the very top deck of the ship. So she is as high up as you can go. She's actually right next to the bridge, which is where the NOAA officers control and drive all of the ship. So she's going to the front, to the bow of the ship right now. But that area is just a nice space that they have some really nice chairs. You can go up there and stargaze at night, or if it's nice during the day, you can go get some fresh air. She's currently in the bridge, so we are in port, so we don't need anybody up there driving the ship. But normally, 24 hours a day, the NOAA Corps officers have shifts that they rotate so that multiple people, at least two, are always on the bridge, making sure that the ship is headed in the right direction, that there's no marine traffic, so other ships that might be coming by, and making sure that the ship is operating safely.
Awesome. Yeah, we don't normally get to get a sneak peek in here because everybody's busy at work. But then I realized, oh, we're at port, so we can see that. And I noticed, Jess, if you turn around, there's like a barbecue grill there.
Yeah, like so this is it's essentially like an outdoor bridge. So this is a small area where they can stand on the other side, and they can, if they need to be checking what's happening on the back deck while they're steering the ship, they can see the back deck from that side bridge. And so that way they can more safely operate if we're working with the ROV or with the CTD.
Awesome. Oh, now we get a nice view of the
ship.
That's how everybody gets on and off. Fantastic. All right, now we talked about some of the things you have seen underwater on the ROV and through the sediment sampling. Now, when you're out on deck, do you ever see other cool stuff at the surface of the water? I heard that you might get a chance to see like whales, sharks, dolphins, things like that. Have you seen anything on this expedition?
We have seen some dolphins. I think we saw some when we were out at sea, but we've also seen some that just come and hang out by the side of the ship when we're in port. So I think we have a video clip that you'll see some of the scientists enjoying their marine mammal friends that have come to say hello in Pascagoula. It was really rough yesterday and rainy, but these dolphins were there to bring a little happiness to our day.
Oh,
There's one jumping,
wow.
yeah, eating. So they're really fun to watch. And when we're underway, they often will ride on the bow of the ship. So you get them kind of going along with the ship, which is really cool to see too.
It looks like it found a fish and it was throwing it around and giving you a little display there of its
Yeah,
fun
they're very
fun
smart.
personality. Awesome. All right. Do we have any other questions before we get to wrapping up? Want to give the folks here a moment. Oh, we have a question. If the cups are shrunk, can you show the kids? Oh, we have one of our friends from USGS had a project that they did with some students where they showed the change in pressure underwater, right? So by taking a styrofoam cup and putting some art on it and sending it down and bringing it back up. Do you have any of those done yet, or you guys didn't do it yet?
I have done them. I really tried to make sure they got done in the few days that we had. So this is a normal size styrofoam cup that often when we're out here, we like to decorate them and we send them to the bottom of the seafloor where the pressure is a lot higher. And so what happens is the air from the styrofoam actually gets pushed out by that pressure. And so the cup shrinks. And so we have some cups from Miss Rain's class that they sent. So they turned to be about this size in comparison. So they are about half size, which is pretty awesome. These went down to 2,600 meters, which is about a little over a mile and a half. So these went down as deep as we have gone on this expedition. And I can kind of show off the ones that we've got from that class. You guys will get these back when we come back to shore. So we will hand deliver these back to Miss Rains, and you guys will get to take those home or keep them in the classroom, whatever you choose.
That's so cool. Are they like harder now since they're more compact?
Yeah, definitely. And we have another one that Nina from our group had drawn. She works for the Smithsonian, so she drew the Smithsonian symbol on there and the coral lab that she works in. But yeah, so they are harder. Like they lose their some of their flexibility when they get shrunken versus the styrofoam, which is really, and you can break the styrofoam, right? If you squish it. These you like truly cannot break anymore. They are flexible forever.
Awesome. I don't know if we have any other questions before we wrap up, but I'll give our friends behind the scenes a moment to see. Lots of diversity. Questions from our class. Do any of your scientists crew use accommodations for disabilities? That's an excellent question. Do y'all, have any experience with that on board?
Yeah, that is an excellent question. So we do, there is an effort to accommodate as much as possible. We do have a scientist on board with us on this expedition who is technically deaf. He's able to hear with an implant, but he has been able to sail for his whole career, roughly 20 years. And in his experience, there's more accommodations here in the US on NOAA ships than on some of the other vessels in other parts of the world. There is a medical assessment that you have to pass to be able to sail, just because you have to be able to move yourself up and down the different decks. There's stairs all over the ship. And then you also have to be able to evacuate the ship if there were an emergency. So go overboard by putting on an immersion suit or a life vest and be able to stay in the water until you can be rescued. So there's definitely accommodations for as much as we can make possible. But it's a physical job to come out to sea. So there's definitely a requirement in order to be safely on a ship.
Yeah. And, something that's really, really special about these opportunities with the telepresence broadcast is we're able to use connectivity to still have other scientists that may not be able to safely be at sea because of the restrictions of moving up and down on the different decks. So we're able to actually have them tune in because of the Starlink connection so that those scientists can still have important input and be part of the discussion and part of the restoration work, even if they're not able to. So it's really amazing for us to have this capability too, to make sure that everybody is able to be involved and they can, watch in real time the ROV feed, they can watch in real time the sediment sampling, and even help direct our science team from shore where they should go next or have input or insight, which is really amazing. So I know NOAA as a whole works really hard to to improve, accessibility and diversity for all the staff to have more opportunities. So that's a great answer. Thank you, Alexis. And I'll just note that Dr. Andy Davies, who Alexis is referencing, is from University of Rhode Island and actually led the whole building of these amazing benthic landers, which are going to be down on the seafloor for about a year, collecting really important information at depth. So we'll actually pull that. Oh, there's a picture of them from Andy Davies' lab at URI. So we'll actually find that article about that and put it in the chat as well. So y'all can check out the amazing work that his science team has done to contribute to this restoration. Awesome. All right. I don't think we have, do we have any more questions as we wrap up? Thank you all so much for your time joining us. I hope this was a little extra highlight since you're stuck at port right now. Oh, hi. Brenna says, thank you so much. We had to leave the stream, but this is very cool and the students loved it. And for anybody that's still online that has, other colleagues, teacher friends, or students that are interested in this live stream, we will have it. It is recorded, so we will be posting that up by the end of the day today, so that we can share it with your other classrooms, your other friends and family, even if they're interested. So it will be available at the same link that you came to for this live stream. So if you share that link, anyone will be able to log on and see this recording later today. But thanks everyone for joining us. Thanks to our team on board and our mobile cam expert, Jess, for walking around and showing off our amazing ship, Nancy Foster. And we'll take a moment here to pop some links into the chat to make sure everyone has an opportunity to visit some more of our pages and learn more about our restoration projects. But thanks to y'all on board. And as we say before folks head out to sea, fair winds and following seas, everyone. I hope you get better weather for the rest of this expedition.
Thanks, Sasha. And thanks, everybody, for tuning in. It was awesome to have a highlight to our day since we're not getting to do our operations.
Well, I hope you still get some ice cream.
I'm sure we will, always.
Awesome. Thanks. See you soon.